Hello everyone, this is Digvijay, KL University, studying BTech Biotechnology. In this video, I'm going to explain you about the restriction enzymes. So, first, let us see what are the contents I'm presenting you in this video. First, history of restriction enzymes, and then introduction about restriction enzymes, and types of restriction enzymes, nomenclature of restriction enzymes, classification, and then at last applications of restriction enzymes. So first, coming to the history of restriction enzymes, in early 1950s scientists noticed some bacteria were more likely to get infected by viruses than others. Over the next few decades, research revealed that these resistant bacteria had a self-defense mechanism, enzymes that cut DNA into pieces and so restricted the virus. These restriction enzymes or restriction endonucleases work by recognizing and cutting specific palindromic sequences. A restriction enzyme is a kind of nuclease enzyme which is capable of cleaving double-stranded DNA. The enzymes may cleave DNA at random or specific sequences, which are referred to as restriction sites. The recognition sites are palindromic in origin, that is, they are sequences which are read the same forward and backward. Restriction enzymes are produced naturally by bacteria. The bacterial species use it as a form of defense mechanism against viruses. In bacteria, restriction enzymes are present as a part of combined system called the restriction modification system. The bacterial species modify their own DNA with the help of restriction enzymes, which methyl it. This particular process of methylation of bacterial DNA protects it from cleavage of its own restriction and endonucleases. Coming to the types of restriction enzymes, there are two types, exonucleases and endonucleases. In endonucleases are the exonucleases are primarily responsible for hydrolysis of terminal nucleotides from the end of DNA or RNA molecule, either from 5' end to 3' direction or 3' to 5' direction. For example, exonuclease 1 and exonuclease 2. Endonucleases. Restriction endonucleases recognize particular base sequences within DNA or RNA molecule and catalyze the cleavage of internal phosphodiester bond for echo RI, BAM HI, etc. And restriction enzyme nomenclature. So restriction enzymes are named with specific set of rules, which is called the nomenclature, and they are an abbreviation of the genus and the species of the organism to three letters. For example, ECHO for Escherichia coli identified by the first letter E, E of the genus and the first two letters CO of the species. It is followed by a letter number of combination of both of them to signify the strain of the species, and a Roman numerical to indicate the order in which the different restriction modification systems were found in the same organism or strain. Coming to the classification of restriction enzymes, there are Based on the types of sequences identified, the nature of cuts made in DNA, and the enzyme structure, there are three classes. First one, type 1 restriction enzymes, and type 2 restriction enzymes, and type 3 restriction enzymes. So here I have pasted a pic, a photo for clear understanding of types of restriction enzymes. Here you can see type 1 restriction enzymes are common but not as much as restriction type 2 restriction enzymes, and these cut both strands at a non-specific location 1000 base pairs away from the recognition site. And restriction and modification, single multifunctional enzyme, nuclease subunit structure is heterotrimer, and cofactors are ATP, Mg2, SAM. DNA cleavage requirements are two recognition sites in any orientation, and these do not have enzymatic turnover, and these have DNA translocation. Site of methylation is at recognition site. Coming to the type 2 restriction enzymes, these are the most common restriction enzyme among three types. These cut both strands at a specific, usually palindromic, and these separate nuclease and methylase. Nuclease subunit structure is homodimer. Cofactors are Mg2 plus. DNA cleavage requirements are single recognition site. These have enzymatic turnover, and DNA translocation is absent. Site of methylation at recognition site only. And coming to the type 3 restriction enzymes, these are very rare among the three, and recognition site is a cleavage of one strand only 24 to 26 base pairs downstream of 3 recognition site, 3' recognition site. Restriction and modification, separate enzymes sharing a common subunit. Nuclease subunit structure is heterodimer. Cofactors Mg2 plus. DNA cleavage requirements, two recognition sites in a head-to-head orientation, and this have enzymatic turnover. DNA translocation is absent. Site of methylation at recognition site. Applications of restriction enzymes. Restriction enzymes are utilized for gene insertion into plasmids during cloning and protein expression experiments. They are also used for SNPs analysis and identifying gene alleles. However, this is only possible if a mutation alerts the restriction site of the enzyme. Restriction enzymes are used for the restriction fragment length polymorphism analysis for identifying strains or individual of particular species. Thank you.